All right, this is going to be fun. But I do talk pretty slowly, so 1.5, 2 times speed probably works out pretty well. So this is very crude work for getting some ideas down about how a very specific group of stem cells may proliferate in response to beta-hydroxybutyrate. That's a lot. So for a bit of history, if you saw my video on planning a stem cell treatment, you may have seen me, or you may remember me showing this study in there. So this research, this was January 2017, official publication date. So that 2017 is when I finished my PhD working in Dr. Radajak's lab in Louisville. I was working on a project not directly on these very small embryonic-like stem cells, but I was working on adjacent research looking at epigenetics of a type of cancer cell that the idea is that they may originate from the same primordial germ cell that very small embryonic-like stem cells are thought to originate from. So anyway, they may share a developmental origin, and I was looking to see if there was something about the epigenetics of the cancer cells that may play a role in why, helping to explain why they proliferate basically uncontrollably, whereas very small embryonic-like stem cells, when you isolate them in a petri dish, they don't proliferate, even in response to, normal growth factors, cocktails, that kind of thing. So yeah, I guess some history, and then why I exactly think that beta-hydroxybutyrate may induce the proliferation of VSELs, and just everything leading up to that idea. So basically, when I was working in this lab, when I originally joined back in 2013, that's when I started my PhD there. These cells were very, there was a lot, you can look it up on Google, there was a lot of controversy emerging around the cells, and there still exists a lot of controversy to this day. But, I didn't know a whole lot about any of this stuff. I just thought that it was really cool that there were these stem cells that may offer an alternative to other pluripotent stem cells, like embryonic stem cells, that carry not only some ethical issues, but also the potential for tumor formation. So I was really excited to see that there was an adult, what they call an adult stem cell, that may offer many or all of the benefits of embryonic stem cells without the danger of teratoma or cancer formation. So all that is to say that I've been pretty obsessed with these stem cells in particular for over 10 years now, and being excited, to work on even if I wasn't working directly with the cells, to work on a project that was related to the cells to help try to understand exactly what could help induce, or what could help explain why these stem cells are what's called quiescent, where they don't proliferate in response to the normal growth factors that can promote proliferation in other cells. Why these cells don't divide in a petri dish, basically. And so ever since then, I've been scouring the research, trying to form my own ideas about what could help explain this, or other potential ways to induce proliferation, or how does this happen in the body? And so anyway, this was a really exciting paper development that happened near the tail end, or was released near the tail end of my time in the lab. And what's interesting about it is that this is, if I remember correctly, this was the announcement that Dr. Radijak and the team had been able to induce proliferation in these cells in a petri dish without the use of basically in what's called a chemically defined medium. They may have used that wordage. Yeah. Chemically defined media free from feeder layer cells. Now, why is this important? Well, they know chemically defined media basically means you know exactly what the components are that are present in the media where the cells are. So the team had been able to induce proliferation, or basically what's called they call them VSELs, derived spheres. So they could expand the number of these cells before, but they did it, I believe, above what's called a feeder layer. I think this is a drop assay. I can't remember exactly all the details. It's been a while since I've looked at it. But basically, what they found previous to this big announcement was that the stem cells, when they were induced to proliferate or divide, it came at the expense, or it happened alongside, you could think of it either way, I guess. It happened alongside an increase in methylation. Of course, I can't find it now. An increase in methylation at what's called a paternally imprinted region of the genome. So this is something called bisulfite sequencing. It's a way to measure the methylated cytosine content within a region of DNA. And these are called, these are the bisulfite sequencing results for specific regions of DNA where they have what's called an imprint, a genomic imprint. And what that means is that there is a specific pattern of methylation inherited, and other epigenetic marks, as I understand it. Specific pattern of epigenetic marks that are inherited from that are present on the chromosome inherited from the mother and are different than the pattern inherited from different on the chromosome inherited from the father. Now, I've been out of this space for many years now, so I may misspeak on some terms, but that's basically the idea. You inherit one set of epigenetic marks from your father, you inherit another set of epigenetic marks for the same region from your mother, and what that can result in is different, these epigenetic marks can help to dictate chromatin availability, or basically what sequences of DNA are available to be transcribed. And what this results in is that from the mother or father, you may have different availabilities for specific genes. So let's take H19 for example here. So H19 is a long non-coding RNA, and it is in I can't remember how large this region is, but it is close to another gene called IGF-2, insulin-like growth factor 2. And so that is a protein coding gene. And you inherit one copy of this region from your mother and one copy from your father. And in what they call somatic cells, so basically adult non-stem cells, or even maybe types of adult stem cells. The methylation pattern is such that this specific area between the genes carries a hypermethylated signature from the father, and it carries a hypomethylated signature from the mother. But in these stem cells, these very small embryonic-like stem cells, this is the bisulfite sequencing results. So this is the pooled results for both chromosomes across many different thousands of cells, I assume, to get enough DNA to do this. But this indicates that the region is hypomethylated on both the paternally inherited copy of that region as well as the maternally inherited copy of that region. And as we can see, I believe they've got the results here. Yeah. So this results in, these are examples of somatic cells. So this is hematopoietic stem cells and STs. What are STs? Probably stromal cells. And I'm all over the place here. Let's see. So, right. So H19, when that area of the genome is hypomethylated, it allows for a 3D conformation of the DNA that basically allows for, it increases the availability of H19 gene to be transcribed when this is hypomethylated. When it's hypermethylated, it does the opposite. It decreases the availability of this gene to be transcribed and increases the availability of IGF-2 to be transcribed. It's a very crude explanation. This is much more complicated. I believe this E stands for enhancer, so there's a lot of things at play here, but that's the basic idea. When this is hypomethylated, this is very available, this is not very available. When this is hypermethylated, this is not very available, this is very available. So when comparing this specific region, H19IGF2, the methylation sequences to these somatic cells, so hematopoietic stem cells, closer to 50, and what that indicates is that most likely the paternally inherited chromosome is hypermethylated and the maternally inherited chromosome is hypomethylated at that region. And same with these stromal cells. Embryonic stem cells are interesting. They're a bit different, kind of in a world of their own, so not really sure how relevant that is for comparison here. But when we look at the gene transcription as basically a proxy for gene availability based on methylation patterns, from this we would expect very high H19 transcription, very low IGF2 transcription. This would be basically these could help set the standard for even transcription from both areas. That does not necessarily mean even number of copies. It's more like these are the controls essentially. So they're closer to 50% methylation, so whatever balance of H19 or ratio of H19 to IGF2, that would be the control. And then the idea would be that the H19 to IGF ratio would be much higher for VSELs compared to these. So that is a lot of very detailed information right there, but that should show up here. Yeah, so compared to the controls, the somatic cells, VSELs have very high H19 mRNA and low IGF2 mRNA. So that's basically, that is at the core, at the heart of helping what the idea is behind what controls the proliferation of these cells. And there's many years of work that the team had put into this, so there's a lot more detail. It's been a while since I've looked at all this stuff again, but anyway, that's the idea. So when these cells do proliferate or divide, and they form what are called, they call them VSELs DS, VSELs derived spheres. We can see the changes in, they put in a graph. So they used percentage of methylation and monitored that throughout the process of deriving these spheres. So you can see that, it looks like they're co-cultured over C2, C12. I think that's a muscle cell line. Yeah, and so you got more cells, and what they could monitor was that the V cells, you can see H19 here, it went from very low methylation to closer and even surpassing the 50% mark. So yeah, so anyway, that's the idea, is that H19, I mean, there are changes in other regions here, but the H19 IGF2 locus, is what it's called, is at the heart of the explanation as to why these cells don't proliferate in culture. And when you are able to induce proliferation or cell division, it is accompanied by an increase in methylation at specifically this locus, at the DMR, differentially methylated region. So that area here, that helps to control the ratio of H19 to IGF2 availability, at least. So all that, now that that's out of the way, so that is the idea behind what controls the proliferation of these cells. So when I was working on my own research, I came across this study had been released. I came across it a bit late, somewhere around 2016, but I became this is where my obsession with fasting really began, if I remember correctly, because if you look in this study from Dr. Longo's group, you actually see that they assayed very small embryonic-like stem cells. Now, they only call them very small embryonic-like stem cells in the supplementary information. I don't know exactly why they do that here, because, so this is figure S2. You can see that they call them mesenchymal stem progenitor cells, MSPC, also known as very small embryonic-like stem cells. So in the main text, they only use MSPC. And what they were doing was assessing whether or not, assessing the number of these cells, or relative number of these cells per million bone marrow cells, in control mice, and then in older mice before and after, well, not even before. I think these are completely separate groups, if I remember correctly. So control, old, and then older mice that had been doing several rounds of fast mimicking diet and had just gone through the refeed period after a fast mimicking diet. So this was, when I first saw this, it was incredibly exciting. Now, whether or not these are only V cells in their in the work they did to the flow cytometry work, I don't know for sure. what they isolated was some lineage negatives, SCA1 positive, CD45 negative. But you can look, if you're interested, you can look in the literature and see that it's not easy to isolate these cells. Groups have historically had trouble isolating these cells. So as far as I know, they don't show the breakdown, all the details of their flow cytometry. So I can't really compare that to other published results from VSELs isolation. So I don't know how pure of a population this is for VSELs, or even if it does have any VSELs. I imagine it would if it is looking at this population in general. But anyway, all that is to say, this helped spur my fascination with fasting. And so where is this going, right? We've got epigenetic imprints that are thought to control the proliferation of these stem cells. We've got fasting that seems to cause, at least upon refeeding, cause an increase in what looks to be the total number as well as the total number of BRDU incorporated cells, which that's a marker for proliferation. I think it gets incorporated into the DNA of dividing cells. It's been a while. I'm all over the place here. So anyway, big takeaway from this study, some group of cells, there seem to be more of them, upon or after doing fast mimicking diet, refeed. Don't know if it just requires one cycle, if it only happens in old mice. Lots of unknowns here, but big takeaway was this may help explain how V cells divide in vivo. So again, I became obsessed with this, and where is this leading? Well, when you go through, actually to back up, let's look at how they were able to induce proliferation in this basically announcement. What they used was something called valproic acid, or VPA. And they say in here, although they don't show the results, which is interesting, we found treatment of V cells by histone deacetylase inhibitor valproic acid remethylated the DMRs of parentally imprinted genes that were erased in V cells. So that was the H19 IGF2 locus. There might be another one. I can't remember if there was another looks like GRF1 is also hypomethylated. So maybe that was also, I don't know, but they don't show the results. So as a result, able to force proliferation of V cells and expand them ex vivo by 10,000-fold in chemically defined media. So VPA is the key here. It is what's called an HDAC inhibitor, so a histone deacetylase inhibitor. Histones are part of the they are a player in the epigenetic landscape. So histones are the proteins around which DNA is wound. So big picture, when DNA is wound up very tightly around these histones, it's much less available. And when it is unwound around these histones, it is much more available. Now, there are other things that play with epigenetics to help determine the availability of gene transcription or genes to be transcribed. But basically, what this is saying is that they were able to change the epigenetic landscape using, or they treated the cells with something that is known to change the epigenetic landscape, and that just happened to induce proliferation, and with that came remethylation of the DMRs. So assuming they mean H19 IGF2 and probably the RAS GRF1. They do talk about H19. Really unfortunately, it had been a while since I've looked at this again. I'm surprised that they did not include the results, the methylation results for these. So here we have an HDAC inhibitor that is inducing the proliferation of these stem cells in chemically defined media, which is important because there's no feeder layer, right? So in here, co-culturing the stem cells above a feeder layer results in cell division and formation of these spheres. That's not a chemically defined media because these cells are releasing growth factors, exosomes, signaling, all kinds of things that can potentially induce changes in cells. And here, it's chemically defined. They know exactly what's going in. So, where am I going? They use an HDAC inhibitor. Well, something else that's an HDAC inhibitor is beta-hydroxybutyrate. And beta-hydroxybutyrate is at relatively high levels in mice that undergo fast mimicking diet. So they even have a comparison table here. But this is they only show the changes. What we want are absolute numbers. Here we go. So supplementary figure 1, S1D. You can see, it just says ketone bodies, so. Okay, so control fast mimicking diet, that must be during the fast mimicking diet, and then during the refeed period for the fast mimicking diet. So anyway, during the fast mimicking diet, these mice have beta-hydroxybutyrate levels on average around 1.75. That is if you have experience with ketogenic diets or fasting, none of this is news. Most people probably never see this level of beta-hydroxybutyrate unless they try to, meaning they make conscious change to their diet to either go into prolonged calorie restriction. Could probably do this. I'm just going to hypothesize. Actually, I know it can. It just depends on the degree of calorie restriction. Water-only fasting can get to this level. Ketogenic diets can get to this level. But a normal, everyday diet, say the standard American diet, people will basically never see this level unless it's a goal. Really, they make a conscious effort. So, anyway, in my mind, I see, okay, well, here's an HDAC inhibitor, histone deacetylase inhibitor, at relatively high concentration that happens during the time, or at least before, that occurs that level is seen before. Why can't I find this? Yeah, here we go. So unfortunately they don't show they didn't assess these values they didn't run this assay on mice that were fasting. It would have been really interesting to see if the value, if if there was any difference between the control mice that were old and control mice during the fast. But anyway, so during the fast, there's relatively high level of an histone deacetylase inhibitor circulating in serum. I can't remember if they did serum or whole blood. But anyway, and then during the refeed period, they see this, what looks to be a relatively large difference in, it looks like total. I mean, it's pretty obvious it looks like total. I don't know if they actually assess total, or if they're just assessing the BRDU incorporation. But either way, big picture is it looks like beta-hydroxybutyrate could be playing a role in the proliferation of these mesenchymal stem progenitor cells, which they called VSELs. Okay, so since then, I've just held on to this idea. There's been a lot of research going on in the background. I'm most likely never going to get into a lab to actually test this out. So now that I have a much better knowledge management system, I don't have to try to remember or put up whiteboards and take pictures of the whiteboards, and then those pictures are somewhere, I don't know where they are. All these ideas have been lost to time. So I began re-exploring this in more, I guess, detail, or just re-exploring this idea, and I wanted to get it documented, basically, because I'd wanted to write some review article about it, never got around to it, so let's put it on YouTube. Okay, so how this could work, because it's just an idea. There has been more work done on the proliferation of VSELs and what can induce that. There's something called UM171 that can do this. One thing that's important to show. From Eric Verdin's group. It's interesting how all these things happened within a few years of each other. So basically this this study demonstrates that if you, the level we were just looking at in here, it's about 1.75 millimolar. Okay, so about 1.75 millimolar on average for these mice at some day during the fast mimicking diet. They probably may have been on day 4. I think these were 4-day FMDs. Anyway, that level of beta-hydroxybutyrate does function as an HDAC inhibitor. So we see a 1 HDAC activity, IC50. it looks like it can induce epigenetic changes in this type of cell, at least, whatever type of cell this was. Yeah, it can induce changes at 1 millimolar, 2 millimolar. IC50. I think that's inhibitory concentration for 50%. So anyway, takeaway is that this level of beta-hydroxybutyrate can be active as to inhibit histone deacetylase activity. Or, I mean, that's what it looks like they're assessing. I've also heard somebody, maybe a Rhonda Patrick podcast, I can't remember, talking about, maybe it also acts as I can't remember what it's called, but instead of inhibiting the deacetylation, it can promote histone acetylation. They may have just been talking about butyrate. I think it's Rhonda Patrick's interview with Dr. Eric Verdin. So it was his lab that looked at this. So all this is to say, this seems to be potentially physiologically relevant as an HDAC inhibitor. So, what was I trying to piece together? Well, to my knowledge, VSELs HDAC inhibition specifically has not been looked at. I'd love to be wrong about that. But to my knowledge, direct evidence showing that the mechanism of action through which valproic acid induces cell division in V cells, I have not seen evidence that, or anyone look to see if it is through HDAC inhibition. I mean, valproic acid is an HDAC inhibitor, but that doesn't necessarily mean that's exactly how it's working in the cells. So I haven't seen mechanistic data that I can think of showing HDAC, valproic acid inhibits one or more HDACs, and that directly causes, say, either the stem cell proliferation, or that causes the increase in methylation at the H19IGF2 locus, and then proliferation happens because of that. So I haven't seen that step, like that mechanistic thing worked out. Again, I'd love to be wrong about that. See if there is something like that worked out. What I have seen, at least the last time I've checked, is that there's another compound that can induce proliferation of V cells. And when that UN-171, and when it induces the proliferation of hematopoietic stem cells, it does so by inhibiting the co-rest complex. Looking at the co-rest complex, what does that consist of? It consists of all these proteins, one of which is HDAC2 and HDAC1. And so this study here shows that BHB is an inhibitor of class 1 HDACs, HDAC1 for sure, and maybe HDAC2 at what's called physiologically relevant concentrations, like from fasting. So how it could work is beta-hydroxybutyrate inhibits HDAC1 and or 2 of the co-rest complex. Thus the co-rest complex is overall inhibited maybe it just inhibits the formation of the CoREST complex, so it can't bind to DNA. It's one idea. And that induces proliferation. So these are all very crude ideas strung together. And, it's definitely worthy of criticism as an idea, because what I've done is, well, I think I've gone about this in a decent way, knowing that this was an HDAC inhibitor and it is functional as an HDAC inhibitor at concentrations seen in fasting. It's, at the scene of the crime, when MSPCs are proliferating or have gotten the signal to proliferate, or it's somewhere around that time frame. And this is an HDAC inhibitor. I don't think it's a huge jump to hypothesize that beta-hydroxybutyrate itself may behave the way the VPA is behaving here, and thus could potentially induce proliferation in VSELs in vivo. But I have had that as an idea, and I've looked for evidence to support that idea. So I've not done what's that called? I've not come in from the other route and looked for arguments against it. So, again, at the end of the day, it's just an idea. So I've been going on now for almost 45 minutes. I'm not going to get as far as I wanted. So this is part 1 in what is likely a multi-part series to try to piece these out. Don't know when the next video will be, but here's another study that has also come out since I've been working on this idea. Another really cool study. Suggesting that beta-hydroxybutyrate can induce a quiescent state. So kind of the opposite of what my idea was here. But as far as I know, they did not look at proliferation in this. Well, I guess they called it quiescent, but they must have. I don't remember. Yeah, this suggests that ketone body signaling induces a quiescent state, whereas VSELs seem to be quiescent by nature, or without the aid of beta-hydroxybutyrate, at least at the concentrations seen during fasting. So who knows? I'll have to dig out my thoughts on this one as well, see how that fits into the whole thing. But yeah, so my goal is to dedicate a multi-part video series to look at each of these studies individually and see if I still think that beta-hydroxybutyrate could induce proliferation of VSELs. Yeah. So anyway, I'm Zach. This is part of my learning journey, and I look forward to digging in deeper with this idea in the next video.